Yeah, so thanks for a great introduction, Brandon. And thanks to all the organizers for giving me this opportunity to talk. Hopefully I can entertain you for the next 1 hour. Hopefully it'll be a very interesting talk. And hopefully it might resonate with some of the work that you guys are doing. So I'm going to talk about these three actually frameworks that I developed during my postdoc here in UT Southwestern to really interrogate these causal relationships between cell morphology on these 3D cells that you guys have mentioned from Lattice Lightsheet or Lightsheet or confocal. The modality doesn't really matter. The key is that we can do computation on these rugged surfaces to relate the local morphology to the signal of interest in a sort of causal manner. Okay. And to get things rolling, and obviously it kind of needs no explanation, is that since the founding of biology, really, from the father of Charles Darwin, we know that form is really function. This is exemplified by Charles Darwin's finches, where, the adaptation, the particular characteristics of the beak is reflective of the diet that the finches eat and of the ecosystem. Now, the way I like to think about it is that the human body is also a sort of ecosystem where the different types of cells, they reside in their different niches and they adapt a peculiar morphology is characteristic of their function. And that's developmentally, but also in disease, right? It's the change in the morphology that's also exemplifies some of the disease. So a very prototypical case is, of course, cancer, where when it metastasizes, it changes its morphology to invade the blood vessels, and then it moves off to a different side, right? Invades. And so the real question is, right, we know that the morphology, of course, is very well established. That morphology is a downstream output of signals. So you have signals coming in extracellularly and through the receptors, it activates these Rho GTPases and all these very well characterized effectors of actin cytoskeleton, right? Rho A, RAC1, CC42, and then to make different types of protrusions that you may be very well familiar with, like laminopodia, pharyngopodia, stress fibers, etc. But what's less thought of in the literature, really, and I think really has only taken off because we have now these really powerful night sheet microscopes, right? Is the idea, can morphology interregulate the signaling? So we think less about now about the chemical reaction cascade, but more about, can the morphology itself change itself and to locally upregulate signaling on the cell? And if it does, under what sort of environment and the temporal context, because shape is not a fixed concept. It's a dynamic concept. So really it's when, where, when, and how. And for this, you need a really computational framework. Okay, so let's think about how morphology could potentially regulate signaling independently of, say, transcription, translation, everyone's, but this transcriptomics people want to sell you. So first of all, there's the surface to volume. Everyone knows about this. If you make a sphere and then you collapse it to a constrict it to an oval, right? You concentrate the signal. Also, you have diffusion traps, right? If you make a sufficiently narrow crevice, the molecule cannot diffuse inside that. So therefore, locally, you can see that you've concentrated the molecular signal in that pocket. And then, of course, it forms the membrane itself forms like a receptor sort of a scaffold, right? It can aggregate these receptor molecules, and how it has curvature sensitive activators. We'll talk about them later. And then it itself is like a physical barrier where you can see that other molecules could scaffold stick onto it, and then itself act as catalyst for chemical reactions. And of course, naturally, everyone knows a lot about the mechanical activation. These are force sensors, right, on that sit in the membrane. Okay, so really to summarize all of this, what you can see is that there's a bunch of range of mechanisms in which the cell shape, and not necessarily just the cell shape, but the components in the cell plasma membrane that effectively act to locally amplify or exclude reactions, theoretically. All right, and the concept is that it can spatially organize locally on the cell surface reactions. Think of this as chemical hubs. And so at what scale these reactions could potentially be decided, divided into different spatial scales. So at the top level, of course, you have the whole global cell shrinking, and you see that evidently you put a cell in a color 3D color gel and it's more spherical. You put it on glass, hard, stiff substrate, it's immediately flat, right? And, this is, and if you use reconstituted systems, you obviously see these nano domains, right? Where, for example, the prototypical RAS organizes cells in these pockets, these nano clusters of reactions on the surface. We can't really image those, right? So we in this Lightsheet community, we're really interested in this micro scale, this mesoscale mode of signaling, where these protrusions are about 1 to 2 microns. And so you could image them in a high resolution, Lattice Lightsheet. And so this gives us an opportunity to combine now Lightsheet imaging and computational framing to assess the impact of how these local protrusions regulate signaling. And we have several wonderful scales. So it's wonderful stories about this that I'll talk about. But a question is, okay, does this really matter? Does this potentially exist? So what I'm going to show you now is these are lung cancer cells, right? And they all have the same genetic background, but when you put them together, you can see on the surface is decorated a bunch of different types of protrusions. So this is sort of already a little compelling. You have the same environment, but they express different protrusions. And we've observed, or qualitatively also observed in a lab, if you label particular molecular markers like actin, PIP2, right, and KRAS, these seem to also congregate into these protrusions, right? So qualitatively, there is evidence potentially for this hypothesis that these microscale protrusions are also correlating or can potentially impact the signal. And back in 2023, with my colleague Andrew Williams, we have this beautiful story published in Nature where we actually show that blebs generate these negative curvature, these dynamic blebs, to organize these molecules, these polymer molecules called septins. And septins are one of the only polymers that can detect this microscale curvature here. And through constitutive blebbing, it actually recruits the septin molecule and it activates this pro-survival downstream pathway to allow the melanoma cell to basically become monoicous resistance. And so they survive the normal detachment process from the membrane. And actually what Andrew's gone on to show, and now he will publish this soon, I think, in a preprint, is that if you look at this in mice, in the xenograft of these same cancer cells, you can identify these invasive front with tumor. And what you'll notice is that the septins organize these punctate patterns at the invasive front. So this is where they're like stressed and they need to survive, right? And then inside the tumor, they organize this diffuse pattern. So this is, again, in vivo sort of compelling evidence that they may serve functional roles. Okay, so the question, the real topic of my talk today is how do we test causality in a complex system? So shape is actually incredibly hard to test causality. And this is because it's a complex system where it's downstream of so many molecular pathways. So in effect, in a complex system, what happens if we normally just knock out a gene X, right? What happens is the rest of the system will compensate. And so what you're really phenotyping when you phenotype this is just the reaction of the system. It's not necessarily the function of the knocked out gene in the unperturbed system. And so how do other disciplines, so we can't really use such a crude sort of perturbation technique to analyze this. So in other disciplines, they actually approach this using perturbation-free causal inference techniques. Okay. And classically, there's neuroscience, and so there's a recent paper where you can actually use neural networks, right, to build the idea of this surrogate and computational brain, and then you can inject signals and perturb it to see, how the signals behave and so on. And in earth science, of course, you cannot affect climate change. I wish we could, but unfortunately, we cannot affect climate change, right? So therefore, there are methods available to look at the time series of the weather patterns and predict, for example, El Nino. And it was a nice case in San Diego, right? Very famous paper by Sugihara is they observe in the San Diego Bay Area the sea surface temperature changes with the population of anchovies and sardines. And so causal methods are being used to apply that to see whether, okay, is our anchovies related to the sardines or is actually correlated to sea surface temperature. So again, this is another example of a system where you cannot affect it, but you need to find some way to find it. And of course, we all know about the stock market for investment. Okay. And so actually in the Toulouse lab, before I joined the lab, they came up with an idea of actually trying to use some of these techniques. So in particular, they use this notion, probably the first notion for observational causality, which actually got the Nobel Prize in econometrics, of Granger causality. And this is the notion that if X causes Y, then that means that if you include the past history of X, it should improve the future prediction of Y, right? It should predict the future of Y. Then if you just have Y alone. And that's basically the concept. And the system that they try to demonstrate this in is the Lambda-Podia system, where you have this protrusion retraction dynamics and you have the Lambda-Podia at the front of this. And this is a nice system because it has all the functional redundancies that we were talking about. And if you see panel C, you have these MDR2-3 VASP affecting F acting and then the protrusions. And so what our experiment have tried is we've seen is that if you knock out one of them, the other two components go up. So you don't really get an idea of what that is. And so the approach that was taken in the Delusa lab was actually to conduct this sort of spatial temporal windowing. So we have a package in where you can grid in 2D these windows, and then you effectively track them, and you capture the Lambda-Podia dynamics in the measurement of a protrusion, a normal protrusion velocity. And when you sample those, and you can also sample the molecular intensity within those windows, and what you end up is you have these nice activity maps, which are these consistent time series in order to perform the Granger causality test. And this allowed them to basically dissect the Lambda-Podia actin regulator hierarchy that's presented in this figure at the bottom and at the bottom right here. And so this framework works in 2D, and if you guys want to interested in reading about it, it's actually published a few years ago in Cell Systems. And the package is called U-Integrate. All these tools are available from the Delusa Lab GitHub repo. Okay. So essentially, if we want to apply this also in 3D, we need to be able to simultaneously also be able to extract these morphological signals. So for example, mean curvature and a molecular activity signal conjointly on the cell surface, but as it moves up and down, right, as the protrusions move up and down. Now, of course, this task is much more harder than just 2D. In 2D, it's effectively you have a piece of string, and of course, you can just divide it into little grids, and boom, you're done with the windowing. In 3D, how do you even begin to unwrap a 3D surface, right? Where is the boundary? There is no real boundary to even flatten, right? And so that's the topic really that I want to focus on really in this talk. Also, I show you a very nice idea of this blab, right? So you look at me and go, oh, this is pretty simple. So in fact, actually, if you put this in collagen cell exhibit a huge diversity spectrum of shapes. So I just show you blabs on this nice globular cortical shape. But in fact, it can have many different, these wide diversity of cortical shapes. And you can also count the blebs itself and not super well defined. They can be prototypically shaped, or they can maybe be little bumps. And of course, you can actually have a mixture of these. I mean, broadly speaking, I just drew this bleb Lambda-Podia phylopoid, but that's actually a mixture. Okay. And for a more relevant case study, we can actually look at cancer cells, cancer and immune interaction. This is a prototypical case where here you see a natural killer cell colored in red. And what it does is when it detects the cancer cell, it engages with it, forming an interface called an immunological synapse, where it sends it's cytolytic, so it sends like cytokines inside to really rip it apart, the cancer cell, right? And something you should hopefully appreciate is that before, right, it recognized the cancer cell, it was in a particular shape. But then as soon as it approaches the cancer, it really changes orientation. And then all it's doing is changing its protrusions, right? So there's an interesting intermarriage between the cortical shape and the surface protrusions in this functional. But the key here I want to sell is cell shapes and protrusions are dynamic and they have different temporal scale, maybe even different functions. That's hypothesis. So to really address this, I motivate this development of these three frameworks. First of all, we need a general 3D cell segmentation tool, because you're not going to sit there or with your grad students and annotate everything and train everything. No. And the second thing is we need to be able to compute on these surfaces. I call a term I call coin and say is surface computing. And then lastly, we need a way to do spatial temporal causal inference, because most of the literature methods to do causal inference all involve already nicely constructed 1D time series. They're not looking at the spatial information flow. Okay, So let's talk about briefly about the general 3D segmentation tool that I developed. The method is actually published on bioRxiv. And it's available already on pip and so on. The framework is called uSegment 3D. It allows you to segment basically arbitrarily any cell. But strictly speaking, it's actually not a segmentation technique. We cheat a little bit. We actually perform 2D slide-by-slide segmentation, and what this tool does is it allows you to combine this in a mathematically rigorous manner to form a single 3D segmentation. And I just found out we got accepted nature methods, so hopefully it will come out in print soon. And the crucial thing that actually surprised me is that this method actually beats out native 3D. And I will show. And why is this interesting is because we actually have 2D foundational segmentation model. Hurrah! Right, so one of the first ones was cell post, and then Meta came along and was like, let's do the segment AC model. And now you have variants of this micro SAM and so on and so forth, cell post SAM and so on and so forth. So this is brilliant because you're not going to go and train 11 million 3D images, right, to get a foundation 3D segmentation. But what uSegment allows you to do is take just 2 3D foundation segmentation model, apply it in X, Y, Y, Z, X, Z stack, combine it into one 3D segmentation, right? And boom, you have your 3D foundation segmentation model, but with no training. That's key. Okay. So that was the objective of this project, was can we combine these 2D instance segmentations into one consensus? By consensus here, let's define this, is that it preserves the input 2D segmentation in the view. So if you then take the 3D formed segmentation and slice it in the orthogonal direction, they just still correlate to your input. That's definition. And actually, this is not such a trivial problem. Because by far the most obvious method to do is, why don't we just stitch it, right? And that's what every algorithm, every framework kind of does, convinces you to do. And so you can, let's take this in 1D to 2D. So in 2D case, you can stitch along all the X segmentations, X direction segmentation. We can stitch along the Y direction segmentation. But the problem is when you stitch like this, right? The X direction segmentation shape and the Y direction segmentation shape in discrete space, in the discrete environment, is not actually perfectly matched. And this is just with this one cell example. So you actually then need to resolve things like, you need to match this, you need to think about, okay, if I want to form one shape, do I take one of these, or do I take the union intersection? So what I'm trying to get at is it's a fundamentally lossy operation, such that your consensus is never really going to preserve your input. Anyway, there are tools out there, which we actually compared to in the paper, right, to do this sort of stitching in 3D in 3 directions. Let's cell stitch 3D cell compose. But they're not really consensus, and they're always suboptimal in a discrete format. But they can be fast. Now I want to introduce the alternative format here, which is, and let's take again the same 2D shape. And can we represent it in a different way? And the answer is, of course, yes. You can represent it as the points, and you can also represent it by a gradient, where the gradient points towards the central axis, right, of the shape. And effectively, you're saying, okay, now let's just not have a binary representative shape, but we just describe it as the points that it composes, is the point cloud, and also its direction. So it's kind of voting for its center. Okay, and the beautiful thing about this is point is a scalar concept, right? So it's naturally decomposable into the 1D directions, X and Y. And gradients, of course, naturally have an X gradient component and a Y gradient component. So it's also decomposed, right? So therefore, you can perform perfect decomposition of this 2D into 1D. And once you've done that, of course, well, to get back the recovery, you can actually, this is a coding that recovers the shape perfectly, because essentially what you do is you run gradient descent on the points along the gradient field. So you can basically cluster them all into the center. And then if you were to apply like collected components or something, you can identify that cluster, right, and then map it back to its original shape. And what's the main challenge of cell segmentation? The main challenge is that when you have multiple cells clustered together in conjunction, you can't really identify it. But if you identify it in the transform space, you can actually perfectly segment very easily, and then you just need to map it. Okay, so how can we, what's the best way to compute the 1D gradients? In 1D, this is trivial. It's always going to be, you just propagate to the center. So in X direction, you just propagate to the center here. In Y direction, propagate to the center here. Okay. So this is a reformulation of the problem from a discrete matching to the problem to just a continuous geometric problem. And advantage is we can also use the gradient to smooth over imputed missing segmentations, because you're always going to get this when you apply 2D slice by slice. You're always going to be great, and then great, and so on. But if you have a continuous field, right? You have a real number, you can just smooth over that. If you dilate basically the gradient, you just go over that and it's fine. And this solves the problem of explicit matching, because you don't actually make a hard decision about where the cell boundary is until after you've done the aggregation in the 3D space. Yeah, and so on. And this is related. Actually, cell pose came very close to this idea. So cell pose, if you look at figure 6 of the original cell pose paper, this is exactly what the idea they exploit, where they take their central diffusion transform, so you make the cell pose segmentations in X, Y, Y, Z, X, Z, and then you can combine them together in this gradient manner that I just described in 3D. What our paper does, that's different from the original cell pose paper, is we put this on a rigorous mathematical framework. And we actually find that the center transform that they use is not optimal to represent the shape. In fact, in 2D, you need to propagate towards the medial axis skeleton, right? And the reason why is because the medial axis skeleton allows it so that adjacent cells do not touch, but also they provide the maximum separation between all cells and still gives the idea of the shape. Okay. And so the optimal combination is specified by a distance transform, which maps to a medial axis skeleton. The problem kind of is the medial axis skeleton is not well defined mathematically, but in U-segment 3D, we make a definition of this anyway. And this is the way, and so the choice of the distance transform we found in this paper is actually it controls the shape prior. So the cell pose example is really only applicable for convex shapes. So what this means is even if you train a very perfect 2D cell pose model, because of the way it propagates the center, it's going to break a very elongated shape. That's one of the reasons why they in turn made the omnipose paper. And so if you want to be able to do this sort of segmentation approach for all shapes, you need to choose this new skeletal representation and compute it in a specific manner. I don't really want to go into details of, it's all in the paper, but essentially this allows you then to be agnostic to the different shapes, convex, branch, and also even for the for thin elongated shapes. Okay, so how the key result is does this 2D to 3D approach compare the native 3D segmentation? I have to thank the reviewers for suggesting this extra experiment. And so what we actually did was we compared this to some classical methods. We compared this to some supervised 3D methods, namely two popular methods, embed seg 3D from Florian's lab and StarDist from Martin Weigert's lab. This approach of training 2D. So the unsupervised 3D is very easy. It's just distance transform watershed, where we make the edge segmentation, invert it, and then just find the seeds and do the distance transform. So this is like the classical benchmark. The 3D segmentation is just you take a 3D model, take a 3D volume, and we try to predict the 3D instance masks. And then this our new approach with the 2D to 3D is effectively you just perform 2D training. So you sample the 2D slices, X, Y, X, Z, Y, Z slices, right? And then you just train your 2D model. So basically just complete 2D model training. And then once you've done training in the inference part, what you do is you apply the 2D model along each XY slice to get a XY slice stack, and then corresponding also in XZ to get a XZ stack, and then YZ to get a YZ stack. So you'll have 3 volumetric stacks of 2D segmentation data. And the numbering and the labeling doesn't matter. And then you pump that through our framework, which is U-segment 3D, and out pops your 3D instance masks. Okay. And here is basically quantitatively an example on the plant cells database. And so I've drawn this along the top row here, you see the 2D aggregated version of the 3D. So these are 3D from 2D segmentation. And then bottom here is the native 3D segmentation methods. And you can see that there's actually not too much difference. And hopefully you also find that worse 2D model to start this because of the convex shape that it wants. is going to be the worst. And then the most flexible, the plant seg, which is actually trained on plants, is going to do the best. And what we found remarkable was if you do the benchmarks, so here the dashed line, here corresponds to the 2D equivalent, the 2D U-segment 3D methods compared to its equivalent 3D. So the solid blue, for example, is the 3D lattice method, and the lighter blue and dashed is the corresponding 2D model of the U-segment 3D. And what you find is that actually for this sort of complicated shape, the 2D model is always perform better, and it can perform significantly better. This is sort of surprising, but if you think about it, it makes sense, because if you can take a really complicated 3D shape, but actually, if you look at it through slices, it's actually very simple, right? Much of it is just circles and convexified, right? And so if you get the 2D right, then and you merge it together, it makes sense. You're gonna incur less error than 3D. So let's do some benchmark. We wanted to find out how does the 2D model performance predict the 3D model performance. And so here I plotted is the AP50 of the 2D model, that trained 2D model. And here is the AP50 after you make a consensus to use segment 3D. And what you actually find in vast majority of models and data sets, actually after the 3D performance is better than what you would predict for just 2D. So it's not necessarily a one-to-one translation, it's actually better than the 2D. So you can actually not get away with imperfect 2D segmentation. And these benchmarks are assessed over, I think, 9 different data sets. And similarly, with x-axis, I plot the native 3D model AP50. And on the y-axis, we plot the 3D AP50 with U-segment 3D. And by and large, you can actually see that they're very, very similar. And what's remarkable is that for the, say, for example, the same data set, right, you can even find a 2D model that will exceed native 3D. So for the same data set, you can always find a 2D model that can even exceed 3D. And most notably in one of these data sets, I think this one, yeah, the 3D just tanked. I think that was a start this 3D model where it couldn't really fit the 3D convex shape. But in 2D, it was totally fine. And we vastly boosted the performance. So there's actually 2 cases here where we couldn't find a model that beats the native 3D. And this is just because at the size that we were using in the cross sections, the cells were just too small. So if you, of course, this approach will fail if the cell that you're segmenting in its cross section is too small to be represented well and is kind of filtered out 2D segment. Okay. But what we're interested in this talk is actually about 3D cells, right? And so if we actually use the cell pose, pre-trained cell pose, and you use this method to make 3D, this is what you get. You find that it doesn't actually fully reconstitute the protrusions on the surface, though it gets pretty much the different placement of the individual cells. And this is a feature of most neural networks, because actually these protrusions in these images are actually tiny, 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 only a few pixels. And so the other component of U-segment 3D is actually putting back the protrusions. So here is the direct consensus, the 3D output segmentation, after you combine the various 2D cell pose predictions. And then we add a filter to improve the spatial connectivity. So now you can see that it nicely sort of smooths things out. And then the real genius of the method is actually to apply this sort of guided filter. What it does is basically like a blend between your image, your input guide image, we call it guide image, and the masks. And what this does is it transfers in one step the protrusions or whatever features of interest you want from your image, guide image, to your segmentation in a locally. And so if you compare this to this, you can see that, oh, the protrusions are all back. It has this snapping function. So those are the 2 innovations we input, which is the diffusion to improve the connectivity and this filter to instantly transfer these high frequency features back. Okay. So I've talked about the 3D cell segmentation, and now kind of the meat of the talk here is just going to be the surface computing, which I'm still revising and writing on. And so what do we mean by computing? I've referred to this a few times now. So I define computing as basically any sort of computational task you would like to do and you need to do in order to get the data in the correct format for the causal testing for time series analysis. So this includes things like detecting and segmenting the protrusions, tracking and registering the movement of the protrusions, taking the statistics like measuring the protrusion height, something that sounds simple, but it's actually pretty annoying. And annotating the ROI, for example. So if I'm only interested in one particular ROI of part of the surface, you want to be able to annotate this. And then finally, the object of this talk, which is you want to be able to perform sort of causal inference between X and Y variables. Okay, these are all what I call computing. And something that I recognized very early on in this project is that performing every one of these tasks in Cartesian 3D is inefficient, it's challenging, and sometimes it's just infeasible unless you're a math genius, right? If you're Terence Tao, maybe. And people have figured this out for a long time, right? I mean, for example, you don't compute like distances in a 3D sphere for maps. You kind of unroll the map into different projections in order so that you can better extract distances and for visualization. And people have done this also, for example, notably in developmental biology, where you take the embryo and then you can unwrap it, and then you can track the tissue flows and cells, cell movement in these unwrapped versions. And then in brain, right, you don't compare the morphological variation of the brain in 3D. What you do is you map it to a standard sphere. So whatever the cohort you have, you map it to this canonical coordinate system in order to compare the variations. You can also do it for tubular structures here, like a developing Drosophila gut that Lowell did recently. And in computer graphics, right, if you play games, the way they actually map the textures onto it is you often cut the surface into these sections, and then the graphics artist will paint in 2D, and then you'll map it back into the 3D. Okay. So this is nice, but it introduces, so this is nice. And to show this, we can actually look at a T cell with micro V9. So hopefully you appreciate on the left side where I'm going to show the 3D protrusions versus these other representations, that the other representations are going to be easy to track. So you can see that the micro V9 is not as simple like a movement. It can actually translocate on the cell surface. And it can also split, which is something I find pretty interesting. Okay. But of course, one thing you appreciate is also in these representations, you're going to lose a bit of the actual geometrical measurement. So this is what I mean by 3D is good. Not each representation is good, right? So 2D this is good for getting a sense of the tracking of these things, but 3D is more better, obviously, to get actual geometric measurement. Okay, and that was a pretty simple example. So working with T cells, actually, if you put them together, these naive T cells, they can actually go much more crazy where they round and they can change the dynamic cortical shape and the protrusions. And basically, our framework needs to be able to handle all of these things. That's what I define by universality. And I like to build universal methods. And so the motivation here is actually to go from Earth mapping to cell mapping. And the innovation here is to introduce this concept of a reference, which represents the protrusion as cortical shape. And then this basically isolates the protrusions on the cell surface. And you can think of this really of, I'm not just interested in a flat Earth. I'm also interested in the rich surface features on the Earth's surface and separating that from the cortical shape. And then you can easily then also map the environment, so atmosphere. So we're interested in mapping the mountains, not just the plains. And the framework I came with is called U-Unwrap 3D. It's a totally generic surface computing framework. And what it does is it's targeted towards these binary segmentation you can obtain from 3D microscopy, and you mesh the binary segmentation. This is why U-Segment 3D was important, to obtain these surfaces and map the signal. Then what we do is we introduce these 5 different representations. Critically, we give you access to these mappings between them. So you can map the original surface into now, this is what we have as the cortical surface, the smooth cortical surface. Smooth cortical surface can be integrated out by mapping it to a 3D sphere or even a 2D image. And then locally, we can reconstruct just the topography, the protrusion specific aspect of the 3D surface. And these are the surface transformations you can do, and these are the 3D volumetric transformation you can do, where you can make a 3D volume and into a 3D topography. The analogy I like to think about is, if you had unlimited resources, you could build a fancy, like a curvilinear sort of laser pointer, laser scan in your microscope. And effectively, you can contour into the cell and out of the cell. And effectively, we're doing that. You can think of this conceptually of doing that, but computation. Okay. So this is, or we all do have a preprint. Unfortunately, it's very old. So it contains very little stuff I'm going to show you next. Published in 2023, so I'm in the process of updating this. However, the code base is up and this is updated, and I do have a set of notebooks if you wish to try out the analysis. So how do we get to these mappings? And we'll spend a little time on this, is we can take the 3D surface. And then what we do is we compute something called a curvature minimization flow. Essentially, it just moves the points to minimize the curvature. So you can see this, and it does this in iterative manner, blah, blah, blah. And so it will go through all these different shapes all the way until it gets to like a sphere. And we can easily find like a stopping point. There are various methods to do this, but essentially you can find a stopping point by measuring the amount of curvature that you compute at each iteration. And so we just find it, and it's is a reasonable reference to use. Okay. Now, what this does is effectively compiles a shape space from your original surface, because actually it's like an onion ring. There are infinite shapes contained within 3D shape. And so if you use any of these as the reference, you're going to get a different definition of a protrusion. This is very important. So use the reference shape that best fits what you want to analyze as protrusion. Okay, but the key is once you have this shape, you can then go directly onto the sphere. So this is how we do it. It's guaranteed by differential geometry mathematics, where it's called a conformal map, where we'll map to the sphere, preserving the triangles shape on here, but not necessarily the area. So it incurs quite a lot of area distortion. In particular, you can think of it as basically inscribing the largest sphere you can find into the shape and then pushing everything into it. Okay, and you can do this as long as your shape is homeomorphic to the sphere, i.e. it's genus 0, and genus is just counting number of topological circles that are in the shape. Okay, and by the way, the deformation process doesn't change the genus. So the input shape can be any genus, and the reference is also going to be any genus. So the question arises, how do you make sure that it's genus 0? And so one of the key enablers of the U-Unwrap 3D that we had to design was this genus 0 shrink wrap algorithm, where you take any arbitrary shape, you first compute a genus 0 alpha wrap, essentially, but this is not very tight, not very impressive. And so the next part of the algorithm is actually going to iteratively deform this, a bit like wrapping a parcel effectively at Christmas, of this initial wrapping paper without breaking the wrapping paper into the little crevices, and voila, you get your final genus 0 shape. So this is how we guarantee you can use any reference shape you would like from that you can compute from. Okay. And that's how we map the sphere. Okay. So once you map the sphere, I mentioned that you have these area distortion, because essentially the longer your protrusion, the more it will be shrunk to a point on your surface. And so a critical part of this algorithm is to engineer an area distortion relaxation. So it basically minimizes this distortion or loss, recovering the actual representation of that feature on the original surface, but on the sphere. So after this, the next part of this, to get the 2D mapping, we just perform standard UV unwrapping. So basically you model this as spherical coordinates, and then you can map 2 axes as the azimuthal, and I forget what the other one is called, angle. And this is called the UV mapping. And something is a convenience feature we introduce in U-Unwrap 3D is, of course, you appreciate if you have a sphere, north and south poles are going to be mapped with the greatest distortion. And the equatorial, it has minimum distortion. So we have we allow you to specify sort of a weight function to map only those surface features of interest into the equatorially. So then you can minimize distortion. That's one feature. The other feature that I realized in the development is we need to actually optimize for the aspect ratio of the UV map. So standard UV maps are 1:2 ratio. Now, say you take this elongated shape and you're unfortunate enough to align it so this long axis was mapped to the vertical axis, then you can appreciate that you get a huge stretching of the surface. And so what UMRAP 3D has is automated differential geometrical objectives, which automatically finds rescaling, optimal scaling factor for the width. So now it looks like this, yeah. Which is good because if you, depending on the operations you want to do, for example, segmenting protrusions or tracking, you don't want to be segmenting a pancake, right? This is beautiful because essentially you don't need to, like other methods, explicitly rotate your 3D shape. I can just design and optimize a weighting function, right? So to unwrap the poles of this Drosophila embryo, all right, so you can cut it along this, what's it called, the dorsal ventral axis, right? So you can map the poles in one map, or you can cut along the transverse axis and you kind of unroll it like a cylinder. It doesn't matter. You can achieve this without modifying anything of the original 3D surface. You just need to specify your weight. And then you have this optimization of the UV aspect ratio. Okay. Hopefully I explained that. And then what you can then do is now you have your laser pointer as a rectangle. Essentially, your shape is mapped to a rectangle. So you can actually perform propagation of this to scan the local surrounding. You can propagate it outwards and then propagate it inwards. And what you do is you can build this topographic mapping. To show the effect of that, what it does is it takes your 3D signal. For example, you can take your intensity, volume, you can take your binary, and then you interpolate it, map it by interpolating into this topographic space. And so this just locally kind of flattens the entire volume. And so you're left with these topographic looking volumes. And we can slice through that. So hopefully you appreciate effectively what it does is it takes the reference and makes it flat. And so everything else is mapped relative to that. And you can see inside the cell like this. Yeah. And then the way we get the topography mesh is we then just remesh just the surface of this to get the undulations. And you can again remap the signals. I just want to point out that it's no, UMRAP 3D doesn't just can map the surface proximal volume. You can actually design very flexible wrappings, propagation schemes. Well, let's say here we have E6.5 embryo gastrulating, mouse embryo gastrulating, and it develops this cavity. So you can specify a segmentation of the cavity in pink here and extract the surface. And you can also extract the outer shape here of the embryo surface, and you can just design a propagation scheme within UMRAP 3D to map just the volume in between the green and the magenta surface. When you do that, you can create this sort of topographic volume here, looking down, that maps here the epiblast. But if you, the critically is, if you look at the cross section, right, it doesn't contain anything of the cavity. It's just mapping between the surface. So I'm just trying to highlight that. Our mappings are incredibly flexible and allowing you to also do this instead of just mapping equal distances into the volume. Okay, so we can show, because it's guaranteed by mathematics, this works on basically any reasonable shape, just not like topological networks, right? You shouldn't do this on something like a network type tree type thing. Okay. So here we demonstrate, of course, blebs on this elongated. We can do these dendritic cells where you have these laminopodia, and then you can have this phytopodia on this branch shape. We can obviously did this for the circular, the T cells or the microvilli. It works also for branch, these multiple hierarchical branches. Of course, your reference will also sort of be branched. And it's kind of interesting because hierarchical branches get mapped, hopefully you might see, into these inner rings. So the base of the trunk branch, the primary branch, is the larger area, and inside it will be encased smaller regions denoting the branches that sprout from the primary branch, which is kind of interesting and something that we haven't followed up, but it could be interesting if anyone has a data set. And then lastly, to show generality, we can take that nice little seed life, the lung cell aggregate thing, and we can map and unwrap the entire cell surface, aggregate surface. So it works for any sort of protrusion type. To show that, we can take our favorite segmentation of cells in this aggregate and then remap it topographically. So then you get a handle on the domains, and you can get a handle on how the local protrusions would look like relative to that. And here is the volume metric scan, where on the left, you see the 3D volume scan, which is kind of hard to see how the protrusions relate to underneath the cell, whereas in the topography, you can see with the boundaries how you're able to associate protrusions into the interior of the cell. So effectively, what you construct with this topographic volume is like a 2.5D projection. It's as if you planarized your curved surface. Okay. So the cool thing is what happens now when you apply this sort of framework to map a 3D complex cell time-lapse image. What actually happens is you can naturally decompose this complex 3D surface into the reference, which is really the cortical dynamics, and only the protrusion cell specific dynamics through mapping to the sphere and to the UV. And to show this in a gimmicky way, right, is actually the topography volume captures now the reference shape. And so the topography volume captures this, but in just a DUV type normal rectangle cuboid space. Now this cuboid space, you can assign any other arbitrary coordinate to. For example, let's just do it for the sphere. So now you can see that the inner coordinate reassign the XYZ mapping to that of the inner part and outer part of the sphere. And then if you now embed your topographic volume into this space, you can actually reconstruct the blurb protrusions on this what is a spherical cortical shape. So this is what I call protrusion transfer. And so you can actually take the exact same protrusions that you saw on this 3D cell and reconstruct it on this in a gimmicky manner on this spherical cortical surface. Okay, now let's back to doing some science. So we can now go back and look at again our favorite NK cell and immune cell video, where we've used USecond3 to segment the 3D surface and map the curvature. Then, because we've isolated, we can compute a reference, we can easily apply volumetric registration techniques to first align it, and then secondly to map everything into just a fixed geometrical shape, but still retain the movement of the protrusions, which is the mean curvature maps, right? And here, that just showing the advantage of a topographic mapping, you can actually configure it so the eventual synapse is mapped right into the center of your image, and then you can view how it touches and the way it develops. It's best to look at this in the 2D view, where hopefully you can appreciate now. We can easily see where the synapses are going to form, and we can now measure and extract and track the formation of the protrusions, the protrusion dynamics within just that synapse region to really understand what's going on. So here, this is just applying an optical flow type tracking because these are very transient, and you can remap this into 3D. So this is not super informative. I challenge anyone to come up with like sort of consensus pattern for this. And so this is a nice segue into the third final pillar, which is this idea of a general spatial temporal causal inference framework. So I developed this very early on. It's published in a NeurIPS workshop. So the framework is called, it's just called rebranded as UInfoTrace now. And the way it works is actually not again, you'll find a theme here. It's not actually a causal framework. It's actually a framework to systematically allow you to apply 1D causal measures that already exist in nature, but into a 2D video format. Okay, so these are the 1D causal measures, and it allows you to apply this now to analyze video data. And what you do when you do this, instead of these optical flow type things, is you can find these information highways. So this image here, shows actually some balls colliding with each other, and what this framework allows you to find is the trajectory, the information flow that these balls came from and the angle that they go. And it's significantly less noisy. So this is actually of a video of cells dividing. It's actually significantly less noisy than optical flow for finding how the cell divides, which in this case is just going outside. To explain briefly how this kind of sort of works is it uses the whole video. And what it does is we consider just the central pixel and a neighborhood of 3x3 window around this. And then we formulate the causal inference to say, okay, I want to find the information, the causal impact of this pixel, of the central pixel to the next point of itself, but integrating out contributions from the neighbor. When you do that, you find basically you can compute causality for each of these neighboring window patches, and the flow is established when you basically effectively take the gradient of this. So you effectively find what is the information across a pixel. Sorry, I went a bit fast, but effectively what this allows you to do is take the entire duration of video to find a more consistent measure of the causality in the video. And here this is shown with a highway video where the cars are coming off. And we do this in a multi-scale manner where you appreciate S equals 1, which is the original image size original image size. You find basically the small cars because the window effectively doesn't have a large field of view. But once you downsample the image successfully, you can also capture the cars now moving at the front. Multi-scale information flow just means we aggregate, we basically compute this at all scales and average it all together to understand both the small scale motion and the large scale motion in the video. The crucial component is if you take this approach. So here, if you did the optical flow tracking and take an average, you get this sort of field flow lines, which is kind of weird and has some artifact in. Whereas if you take the information flow, it's a lot more meaningful. You can actually see that what happens is the surface protrusion set up this retrograde flow. So this retrograde flow is important for the establishment of the immunological silence. And this is revealed very nicely here. This information flow. So I've shown you how you can apply causal inference to this, and to show it in a different manner, we can revisit our example of the blebs and the septin. So what we can do is we take segment protrusions in the topographic view. We can map it to the 3D Cartesian. We can map it into 2D. And then the beauty is that in 2D, all these signals, the segmentation, the curvature, the septin are all aligned. They're all already spatially registered. I didn't have to do a second spatial registration. And then what we can do is, if I play the video here, is we can do this at all the different time points. And then in 2D, what we can do is we can use just a bounding box tracker. So we can identify these different curvature points as the blurs, and then track them using your favorite 2D tracker, and then remap it again to 3D and make measurements. And what this allows us to do is revisit our hypothesis that successive blebs are required to accumulate a stable septin population by reconstructing a profile of the mean bleb that we track. So here is the profile of the mean bleb area. And we take a window relative to the maximum time. And then conjointly, we can sample a normalized measure of the septin intensity of the bleb, i.e. in the necked area of a bleb, in the neck of the bleb, and on bleb, meaning it's on the side of the bleb. Now, curvature, what happens is what happens with septin is that they are recruited at a negative curvature, so you'd expect more of the bleb, and that's what's shown in yellow here. What you hopefully see is that there is an incremental increase of septin levels prior to a bleb and after a bleb. And so what this shows you in a quantitative manner is that multiple bleb cycles are indeed necessary to presumably recruit enough septin to form these stable scaffolds. Okay, just to give you a quick glimpse into some sort of idea of future work, is actually you can, UOnRep now provides a very powerful interface to look at tissues in a new context. So we can take the invasive frontal tumor that we have in the septin, we can segment every cell in there with U-segment 3D, and then we can blow everything apart just to show that there's a diversity of shapes in this, even in this small volume. What we can do is extract, for example, a single volume, and it'll be in a single subvolume, and you can see that it will be in contact with multiple different cells. And what's beautiful is now that you unwrap 3D, we can take all the cells that is sort of in contact to, and unwrap the cell surface, unwrap that cell surface, that cell, right? And you can identify the different contact maps. So I would call that contact map. So you can mark the regions of the cell surface in contact with distinct other neighbor cells, and to analyze the spatial distribution of a pattern of the protein expression in these contact surfaces. Right. Whereas currently in spatial transcriptomics, you normally take a bulk expression. You think of all the signals as bulk and compute a mean signal. Here, this really allows you to address what is the spatial heterogeneity of that expression pattern. So not all signals are expressed uniformly, cytologically, they can form puncti, you don't know, right? And this now gives a way to access that in situ images. Okay. And so hopefully this is the end of my talk. I've convinced you I've established these three pillars, three frameworks, which will allow you to now analyze any 3D cell images you like, to test for causality between morphology and the signaling. So they are U segment 3D, U unwrap 3D, U infiltrates. And the work is not finished, and we have a number of hopefully other stories later on in the winter. But that's the end of my talk. And I finished to thank the Uber Labs, and much of this work would not be able to happen without this interesting agglomeration of many different labs together, and also many of our friends from other institutions which have contributed data, and of course the funding from the U54 and other sources from NIH. Okay, thank you very much.
Excellent. Thank you for that, Felix. I would say we have some time. If people have some questions, feel free to unmute and ask your question, or I can go through the chat and see if there are any that people had already asked. Let me see where we're at here. Looks like maybe I see one from the start here. So Vad asks, is the 2D representation required in order for us humans to better perceive the 3D volume and related to the constraints imposed by 2D media like paper and screens?
In effect, yes. If you can map the 3D into 2D, it's easier for us to comprehend time lapse data, because then just the one thing changes and not multiple things happen. But I would say also the 2D representation flattens too much of protrusions, so you can't really appreciate, for example, from the curvature heat maps, how this thing actually looks in 3D, and you might actually get confused because imagine you have a ripple, you can have multiple points of high curvature, right? And so you don't know whether it's part of one thing or part of multiple things. And that's why I think also the topography is a nice way to understand. It's a bit like contour maps, right? And if you go hiking, you won't want to see the contours as well.
Excellent. Looks like a question from Adam. Go ahead.
Yeah, I've got kind of a follow-on question to that. So when you're deciding to segment a particular structure, how do you decide what the boundary is? So you gave examples of blebs and Filopodia, and Lamellipodia. Each of those has a unique kind of spatial frequency shape to it. And at some point, you've got to make a decision of, in your segmentation method, in 2D or 3D or all of the math there, what defines the limiting boundary of that?
That's right.
I'm wondering if you could comment a little bit on how you've approached.
That's right. So you're actually preempting a future paper that would address exactly this question. But I can give you the sketch out the idea. The idea is basically you can use these deformation to optimally find a reference surface, and then we can measure just the protrusion height, such that if you do a mean threshold, that gives you everything that's protrusive on the surface. And then what happens is you can measure the seeds of these individual protrusions to find connected components on the cell surface. And I showed only 3D mapping of closed surfaces, but actually unwrap can do the same thing, but on open surfaces. So then what you do is take these direct connected components, map them into 2D, and then we actually recycle our U-segment 3D philosophy, which is you can engineer now a curvature gradient or a protrusion height gradient or a shape gradient. and then it basically propagates the point as a flow on these unwrapped to the connected and then put it back onto the surface. And that way you don't need to decide what is the boundary. Perfect.
Super, thank you. It was a very nice talk too.
Thanks.
I think we have a follow-up question from Andy here. What is the thickness of the pathology slide for invasive front on the melanoma, and 2D or 3D based on the thickness?
Yeah. So sorry, I didn't clarify. That's not actually a pathology section, so it's not like section by 5 micron or so on and so forth. It's actually just a clear tissue volume. So the whole specimen, the biopsy would be cleared out. So it's actually full 3D volume imaging. And so we just segment in 3D. But otherwise, you're absolutely right. If you have these 5 micron thing, essentially you would only be able to really do 2D segmentation and then try to stitch it together.
I have a question here. In terms of the models that you use for choosing the segmentation, you had mentioned SAM or CellPose. Which ones do you typically use and find the most successful there?
So that's a bit of a nuanced question. So it actually depends. So it actually depends on the markers. So typically we have a membrane marker or a cytosolic marker, right, for the cell shape. For cytosolic markers, actually the best approach is to apply 2D binary OTSU. And the reason why is because neural networks is normally trained with like a mean squared loss. And what happens is that effectively smooths out protrusions. So the binary OTSU gives you better starting shape. And then such that when you apply the guided filter, it's much more accurate, and we have this now. But for where the neural networks really excels is for these membranous stains, where the inside is hollow. So what the neural networks learn is a prior, as a shape prior for these things, such that if it sees part of the boundary, it's able to impute the whole cell, and this allows you to extract the proper surface. Whereas a binary OTSU based on just the intensity threshold will just leave a hollow thing. Right. But you can actually just combine both of them, so there's no reason why not. So you can actually do a consensus for the CellPose or whatever neural network. You do a consensus for the binary, and then you can merge them together, and then use the guided filter on the combine to fix any incontinuity. So that's the flexibility of this approach.
Okay, very good. Awesome. Any other local questions here? All right. Well, thank you so much, Felix, for this.
Thank you.
I learned a lot. So thank you.
Okay.
And congratulations as well on the paper being accepted.
Thanks. Okay.
Thank you very much. The talk was very wonderful.